It is the flow chart of a mechanism of cell injury. See, the most common cause of cell injury is ischemia or hypoxia. It occurs due to less blood supply. Ischemia or hypoxia leads to decrease in ATP generation inside the cell. That will lead to three things: 1, 2, and 3 things. I haven't labeled, but you also here you have to mention here 1, 2, and 3 here. The first one is there is decreased synthesis of membrane phospholipid. As there is decrease in ATP, there is decreased synthesis of membrane phospholipid. There is increased cytosolic calcium inside the cytoplasm of the cell. Okay. Then the third one is toxic oxygen free radical are generated. This is the main thing, that increased cytosolic calcium that leads to reperfusion, means in the mitochondria there will be increased cytosolic calcium level. Generally there is less calcium, but in the case when ischemia occur, there will be decreased ATP that will lead to increased cytosolic calcium, and that will reperfuse, means inside mitochondria the calcium will get entry and increase the cytosolic mitochondrial calcium level. That will activate two enzymes, phospholipase and protease. This phospholipase, name suggests phospholipase, means it break down the phospholipid as we apply in the case of enzyme. This enzyme phospholipase, they break down the phospholipid which is present in the cell membrane. Cell membrane is generally consist of phospholipid only. Our cell membrane, here phospholipid break down this phospholipase break down the phospholipid into the breakdown products, lipid breakdown product, and decrease the membrane phospholipid. And the protease enzyme that causes cytoskeletal injury that leads to membrane damage. All these things lead to membrane damage. Increased cytosolic calcium level leads to reperfusion, and increased cytosolic level, calcium level of mitochondria that leads to increased activity of phospholipase and protease that leads to decreased membrane phospholipid and lipid breakdown product. Protease activation lead to cytoskeletal injury. This all things lead to membrane damage. Here different case, here the toxic oxygen free radical leads to lipid peroxidation, DNA damage. Here DNA damage lead to membrane damage, and all this membrane damage break down the membrane, and there will be liberation of intracellular enzyme. If membrane will be break, the inner content of the inside the membrane will now go out, because the membrane is destroyed. Membrane will protecting the all the ingredients together inside the cell. When the membrane is broken down, there is a pathway to escape. That's why there will be liberation of intracellular enzyme. That will lead to three changes: nuclear change, cellular change, and serum enzyme estimation. Nuclear changes are pycnosis, karyorrhexis, and karyolysis. Cell death. In cell death, Mylin figures are seen. In serum enzyme estimation, SGOT is seen. In nuclear changes, three changes: pycnosis, karyorrhexis, karyolysis are seen. Pycnosis, there will be compaction of nucleus. In karyorrhexis, there is fragmentation of nucleus. Nucleus will be fragmented, and in karyolysis, the nuclear fragments get dissolved. There will be complete disappearance of the nucleus. Pycnosis, karyorrhexis, karyolysis. This is the first step. Next video we will see the mitochondria, calcium, and free radical injury in depth.